So yeah, thanks to the organizers for giving me this opportunity. I've had a lot of fun this week. So there's actually going to be two long tails in this talk. So I'm going to tell you a story about bacterial population growth in a mouse. So one of the long tails will be interested in the distribution of population sizes of different groups of bacteria inside the mouse. And so one long tail will describe the long tail of that distribution. But by the end, I promise I will show you some results where the bacteria were actually injected into the tail of the mouse. And you'll see why that might be different or important. Okay, so this was work done from my group by Matasem El-Gamel. And he was a PhD student. He's now a postdoctoral fellow at Emory University. And it's a collaboration with the experimental lab of Amir Mitchell and his student, Sirkin. So this is the outline of my talk. I'm going to start by showing you some math so that we are all on the same page on what kind of extreme events are going to be important for my purposes. And then I'll talk about the mouse and the bacteria and see how that math is important for how we model that system. Okay, so I'm showing something very simple here. This is a birth-death process. I'm also revealing immediately that I'm a physicist because I do my time derivatives with a dot. It should not be obvious to you why this is relevant for bacterial growth. Bacterial grow exponentially. But I promise we'll get there. So right now, we just have a process where the growth rate is a constant and the death is spontaneous, so the death term is proportional to the population size. And this is very well behaved. It has a steady state, alpha over beta, etc. But we want extreme events. So I'm going to introduce noise to this birth-death process, and I'm going to do it in one of two ways. And I'm going to imagine introducing noise to either of these two rates. And so I'm introducing it in a Langevin form where the strength is this parameter sigma. Eta is a Gaussian white noise variable. Now, if I introduce it into the birth rate, it's very different than if I introduce it into the death rate, because in the first case, it just becomes a Gaussian variable added to the equation. We call that additive noise. In the second case, it also gets multiplied by this n, and so the strength of the noise is proportional to n. We call that multiplicative noise. Okay? Or at least I do.
Does alpha depend on n?
No, alpha is a constant.
So it's an immigration rate.
It could be viewed as an immigration rate, exactly. Okay, and the consequences are very different from the kinds of trajectories that you'll see, right? Additive noise is sort of well-behaved. It's Gaussian distributed around the mean. When the noise is multiplicative, you get these very long excursions from, I'll say, the mode of the distribution, and that is what I'm going to refer to as the extreme events in this context. Okay. And you can ask what is the distribution in each of these cases, and for that you can transition from the Langevin equation to the Fokker-Planck equation. And again, in the additive noise case, this is very straightforward. The steady state solution is a Gaussian. And again, you get sort of symmetric fluctuations around the mean. In the multiplicative noise case, even though all I've done is added this n inside of this sort of diffusion-like term, the steady state has a completely different functional form. It has this weird like e to the minus constant over n, and then it has this power law on the bottom. And indeed, it has this power law is the source of the long tail. Okay, so you get this very long tail corresponding to these extreme events. Okay, first long tail, check. We're all on the same page? Okay. All right, this is a bit of a weirder distribution, so you might be curious about its statistics. Its mean is still alpha over beta, exactly the same as the steady state of the deterministic process. But because of this long tail, its mode has been shifted away from the mean, or you could say the long tail is shifting the mean away from the mode. And in fact, the mode is the birth rate over not just the death rate, but the strength squared of this Langevin noise term. Okay? So in that sense, the noise is playing an effective role of death in the system, an additional role of death. It's contributing to the death. And the intuitive reason for that is what some people call negative drift. I think somebody, Jinsu, maybe mentioned negative drift earlier in this conference. So what is meant by negative drift? Well, if you can imagine that you're on the n number line and you start with noise kicking you up and then kicking you back down, you will not return to where you started, because once you get kicked up, remember the noise is proportional to n, so your downward kick is stronger, and so you end up a little bit to the left, right? So you say, okay, well, what about the reverse process, right? Instead of getting kicked up, maybe you get kicked down first and try to go back, but now your kick is smaller because you're at a smaller end, so you also drift to the left. So on average, this multiplicative noise will drift you to the left, so it's kind of like playing alongside death there. And so you could ask, do we even need death? So in some sense, the answer is no. You could just not have death in the system and have noise play the entire role of death. And that's equivalent to sending beta to 0 or up here epsilon to 0. You get a fully normalizable distribution. It's completely well behaved. The mode is just alpha over sigma squared now. The mean blows up, but that's not so weird for distributions with long tails. Okay, so no death. My answer is no problem. And in fact, this stochastic trajectory I showed you before was exactly this case, where I didn't even have death. And we can see immediately then that this long tail is characterized by a power law, an inverse square power law. Okay. All right. So in summary, additive noise in this birth-death process gives you a Gaussian. Multiplicative noise gives you a long-tailed distribution. In this limit, it goes like 1 over n squared. And I'm showing you in a log-log so you can see the straight line of slope -2. Here I'm also showing you the cumulative distribution. This will become important when I talk about data for the bacterial growth. And in fact, I'm showing you what is, I guess, called the complement cumulative distribution. It's the probability of observing a population larger than n. So it starts at 1 when n is 0, and then it sort of goes down in this controlled way for the Gaussian. And then for the multiplicative case, it's also a power law at the end, but because I've integrated, my n to the -2 has become n to the -1. Okay? There's one last thing I want to sort of introduce before talking about the bacteria in the mouse, and that is that instead of the complement cumulative distribution, sometimes statistics like these are plotted in terms of the rank and the frequency, and I will tell you what that means in a second. But I want you to notice right off the bat that a plot of the frequency versus the rank is basically just a reflection of this complement cumulative distribution. Okay? So what do I mean by frequency and rank, and why is it a reflection? I'm going to go through that with you in a simple example because it took me a very long time to understand. So, and it also is related to how I'm going to show you our collaborators build the data from samples of bacteria. So abstractly, I want you to consider that we have a system, and in that system are samples of populations, right? So here I have 4 populations, each of a certain size, 5 individuals, 3 individuals, etc. So I could construct the population, the probability distribution from this system. You see that 2 of the populations are size 3, so there's a 50% chance of observing n equals 3, 25% chance n equals 4, etc. And then I could do the complement cumulative as well. So when we think about rank and frequency, it means the following. Imagine that instead we recognize that there are 15 total individuals, and we ask what frequency each population out of that total represents. So this one would represent 5 out of 15 frequency, 3 out of 15, 4 out of 15, 3 out of 15. Okay. And at the same time, we know that there are 4 samples of population, and I could rank them by how big they are. Right? So this one gets rank 1, this one is second biggest, rank 2. These 2 are tied for 3 and 4, so I give them both rank 4. Okay. And the important part is that if you think about it for a second, or months in my case, you can notice that the frequency is just proportional to n, the sample size, or the population size, And the rank is proportional to this complementary cumulative probability. Okay? Which means a plot of R rank versus F frequency will look exactly like this, just with scaled axes. And for whatever reason, people usually plot F versus R instead, and that's why it's a reflection. Okay? We're all on the same page? Just like 3 different ways to look at a distribution. P, C, or rank versus frequency. And that's why you get this. And I just want to point out that this multiplicative case I've been talking about, if it were P, it'd be 1 over n squared. If it were C, it'd be 1 over n. And because a reflection of a 1 over a variable is still 1 over a variable, then in frequency versus rank, this would be a 1 over r type power. Okay. All right, so that's going to be relevant for the next part. So now some biological function. Okay, so as I said, we're going to study bacterial growth. The function is growth, and we're going to be interested in how do bacteria grow inside of an animal rather than in a test tube. So here is my mouse, and these syringes contain different populations of bacteria, which I'll describe in a moment, but they're a rainbow because each population is like a different color. My mouse has these 2 lumps on it, which unfortunately are tumors. So why does the mouse have tumors? So I'll also tell you that in a second. But just to make clear what's happening here, we have 5 million total bacteria in each of these syringes. And a cell is called CFU. That stands for colony forming unit. And it's a fancy biology way of saying that it's a cell and it's also alive. If you were to put it on a plate, it would form its own colony. So it's a living cell. So whenever you see CFU, you can think cell. Okay, why are we putting this bacteria on a tumor in a mouse? Well, I didn't know this, but until about 10 or 15 years ago, we didn't think that tumors had their own microbiome with bacteria living inside the tumor. But since that time, it's become clear that many, many different tumors, mostly solid tumors, do have microbiomes. And it's very interesting. I mean, it was essentially assumed that they didn't because it's such an area of high, immune cell activity, that they, were thought to be cleared, but that's not the case. And in fact, if you look at the same patient at the cancerous part of a tissue and the non-cancerous part of a tissue, they can actually have different community structures. So they can actually have a unique microbiome. So all of that is very interesting. It's raised a lot of questions in the biology and medical communities about how the microbiome might affect the cancer progression in patients. And at the same time, for sort of more fundamental-minded scientists, it's raised the inverse question. How does the tumor microenvironment affect bacterial growth? And that's the question I want to focus on for the rest of the talk. So I'm going to ask, we asked, what governs the bacterial growth in the tumor microenvironment? Specifically, how is it different from, controlled growth in a test tube? So to answer this question, this is what Sirkin did. So he used a technique called DNA barcoding. So that just means that in each cell, there's a little piece that's been inserted in its genome that is like a tracking code, a piece of DNA that tells you the identity of that cell. And he made a library of like 40,000 of these little pieces and distributed them amongst the 5 million bacteria. So you can do the math, and on average, that means that there's, about 100 cells that belong to each of these barcodes. So that's what I'm illustrating with the different color. What's important about that is with sequencing, you can tell then the relative proportion of each of these subpopulations within the entire population of bacteria. And it's also important because it's in the DNA, so it's inherited. So if a cell divides, then its progeny also has that barcode. So it's a way to sort of track subpopulations in a big ensemble as they grow. So he took barcoded bacteria. As I said, he injected them in a tumor in mice and then collected them after a certain amount of days and then grinded everything up and sequenced the bacteria, everything, just looking for the bacterial DNA and in particular the barcodes. And from the number of hits, you could tell how many cells were there from each subpopulation. So I'll sometimes call them subpopulations or clones or barcodes, but that's what I mean. Okay, everything clear?
So,
Yes.
mutations won't change like the barcode?
They would, but there were none. Yeah, you can go back and check. Are there any mutations? They're very rare on this time scale because it's, one,
Short
3,
enough.
7 days. Yeah, it's tens of generations, but it doesn't really happen. Good question. Okay, so what did we see? So first I'm showing you what was put in. And again, this is frequency versus rank for these 40,000 different subpopulations. Okay, and this is in descending order. But the thing to notice is that this is mostly flat, right? Sirkin did this by design. So on average, each of these subpopulations is about 100 cells big, so they represent roughly equal frequencies in the total population. And as a control, you can first put it into a test tube, you dilute the whole thing, and you let it regrow, and you see how those frequencies change. And they don't change very much. This is what you get out. There's a little bit of a scatter because, of course, it's not perfect. You regrow with some noise. But this noise is well explained by just a birth process with demographic noise. Okay. Instead, if you put the same sort of distribution in a tumor and you collect, then even after one day, you get something like this. Right? So it's completely much more noisy, right? This is a log scale, in fact. So it looks like it's been completely randomized. And so it looks a little bit hopeless right now, but in fact, what you can do, and this is much more informative, is you can take the collected data and you can do a new frequency versus rank plot, but now the rank is the rank of the new data, right? You can re-rank. And if you compare that to the old data for the test tube experiment, you get basically the same thing, which makes sense. You've created the library in liquid media, you regrow it in liquid media, you get the same thing. If you do that with the tumor data, you get something very different. Yes, Krisha?
I'm a little confused. Are you the x-axis on the two?
It's ranked for each one.
For each
Yeah,
one,
yeah,
so it's
exactly.
different axes. Okay, good.
Two different axes.
Yeah.
Sorry. Yeah. So you get something very different in the tumor, and you get that same different thing no matter which experiment you do, which side of the mouse you use, no matter what day you collect. Right. So it's very robust, and of course, it's this frequency goes like 1/r that I showed you before. Okay. Some of you may know this as Zipf's law. Have people heard of Zipf's law before? Okay, so there's many things that have been seen to obey Zipf's law. Frequency goes like 1/r rank. There's just as many mechanisms to get Zipf's law. And so our next question was, which one is at play here? Can we convince ourselves that some classical models of population growth are sufficient to give this kind of behavior? And so we wrote down what is essentially a consumer resource model, where we have 40,000 different populations, N sub I, and one resource. We know there's not 40,000 resources, so we simplified our lives and said there's only one. Okay, and the populations are growing exponentially. Okay, and the exponential growth rate has two components. One is from consuming the resource, and this is a saturating function that is due to Jacques Monod. This is just basic bacterial consumption of a limiting nutrient. And we have noise in the growth rate. Okay, and again, because it's exponential growth, it's multiplying the population size, and therefore it's the multiplicative noise we suspect that we need for this dramatic power law. And then the nutrient just gets consumed by all the populations, and it gets replenished at some rate d according to some reservoir S star, which is the concentration in the rest of the mouse, for example. So we simulated this model, and we at a given time we took all of the population sizes and we plotted it frequency versus rank, and it's looking pretty good. The dashed line is Zipf's law, but if you wait enough generations, then it sort of falls apart. And in fact, what's happening here is that almost all the clones are going extinct, except for a couple. There's only 10 left at this point, and there is a good reason for that. Those of you who think about consumer resource models may know this reason. It's called competitive exclusion. So it says that you can only sustain as many populations as you have nutrients, and we had one, right? So this model is guaranteed to leave only one surviving population to take over the whole system, and it's just whichever one gets luckiest over its lifetime with the kicks due to the noise. Okay. So instead, in the data, we have like 10,000 surviving populations. So how is that? And to give us a hint on the answer to that question, if you just look at how many surviving populations there are, not how many cells there are per subpopulation, but how many subpopulations as a function of time, you see that that number drops almost immediately and stays roughly constant. So you go from 40,000 clones to 10,000 clones and stay around there. So that suggested to us that this mass extinction happens right at injection, and that's called an infection bottleneck. And so that suggested a hypothesis where most of the cells in your total population die, and in fact, many of the clones go extinct immediately. But if you're a clone that does not go extinct, you are survived by just one cell. And then you regrow based on that one cell. And because you're in a solid substrate, the tumor, then your progeny are likely to be right next to you, and therefore you're competing for nutrient with your own subpopulation much more than you are with other subpopulations. And so you've essentially formed a spatial niche. And so how do we add that to our model? We didn't want to do a full-blown spatial model, especially because this is just sort of a hypothesis. But there is a very simple way to add local limitation by your own subpopulation into this model. And that is due to another French person, 10 years after Monod, who observed modification to Monod growth empirically at higher bacterial densities. His name was Comtois. And his empirical modification was that the half maximal coefficient on this Monod growth law depended, in his case, on the population size itself. And so we made that modification to our model. And the first thing you can notice is that when n is very large, you can see it overpowers this S term, and you just have an n on the denominator and n on the numerator, and I get back to my constant birth multiplicative noise case that was the subject of the first part of this talk, right? So this is an immediate clue that we're going to get the right statistics, and in fact we do, right? So now it's not the case that everybody goes extinct anymore, and we get this very nice 1/r frequency versus rank. So what's happened here is by self-limitation of growth for large clones, you prevent the extinction of the small clones, and you turn this consumer resource model into this very simple case of constant birth and multiplicative noise. Okay, and you can do the comparison in a bit of a more calibrated way. So each of the different experiments had a sort of its own infection bottleneck, a certain number of clones that die. And if you start the simulation with that level of infection bottleneck, then you get this sort of spread of different statistical distributions that match the experimental spread pretty well. And in addition to the statistics, you can get the dynamics, right? This is the sort of 1, 3, 7-day dynamics from the experiments, but in our model, you can sort of average many stochastic trajectories, and it obeys pretty well. You see that, you get this huge bottleneck where the total number of bacteria falls off, and then it recovers. Meanwhile, the number of subpopulations falls off and stays constant, and that seems to be consistent with the data. We don't have the time resolution to see this fall off, but other groups have at short time scales. And so we're basically left with two predictions that could in principle be tested by future experiments. One, I told you, we hypothesized that this is a situation where bacteria form these spatial niches. If you could actually cut up the tumor and see sequence-like bisection, you could test this hypothesis. And also, in case it wasn't clear, what's happening here on a fairly short time scale, like 30 minutes, the lifetime of the bacterium, you're getting like exchanges of who's the top ranked, who's the biggest population is always sort of constantly turning over in time. So we have to kill the mouse to get every time point, but if you didn't, you could ask whether the dominant clone is always changing. And that's beyond my expertise. There might be ways to do that, I don't know. Okay, last thing, I promised you the second long tail, right? So indeed, the first thing they did is inject the bacteria into the tail. They still collected at the tumor, but they injected it in the tail to get it in the bloodstream. Any idea on how the results might differ?
Now it's additive.
No, it's not additive. This doesn't change the competition mechanism once they get, so I don't think so. But good guess.
You don't have the spatial.
So the bottleneck is much bigger, right? It's just far less likely you'll even get there, right? So it's just a much bigger infection bottleneck. So instead of 7,000 clones surviving, there's like 100. But interestingly, the total number of bacteria still reaches about 1 billion, which is about the same. There's a tumor carrying capacity for bacteria. You can't really resolve the power law anymore, but we think that's just because you have limited decades of resolution. We think the mechanism is unchanged. Yeah.
But in that case, wouldn't you recover because if they are into clustering, maybe they have more resources and you lose the special component, no?
I think you would amplify the spatial component because if only 100 clones are present, it's definitely only one cell is seeding each clone, and then they just have a bigger space to grow. So you get larger patches on the tumor. That would be my hypothesis if our model is right. Yeah. Okay, so I talked to you about multiplicative noise and these extreme excursions. I showed that it well describes bacteria in tumors. And that's due to a model that includes an infection bottleneck and local growth limitation. And it essentially reduces to this type of model. And so with that, I will just acknowledge especially Mata-san again, and thank you all for your attention.
Colin? Yeah, thanks, Andrew. Really nice talk. I noticed in your models with multiplicative noise, it looked like you took a Ito interpretation every time. Can you comment on why you did that and how it might be different if you did another interpretation?
Yes, that is a good question. I am taking an Ito interpretation. I think I can't remember if this is a case where if you took a Stratanovich interpretation, you would still have the same power. You would just shift the location of the mode. But I'd have to think about that. I mean, we could talk later. Yeah.
Back.
So the multiplicative noise gives you this long tail, but you could also just have additive noise with a long tail and death. Have you thought about or looked into how things would change? I don't know, to make sure that there's not an alternative explanation.
Yeah.
I just have long tail. Like, is the multiplicativity or is it the long tail that gives you?
So I'll say, since you mentioned death, it's a bit conspicuous that there's no death in this model. Essentially, noise is playing the role of death, right? Of course, if eta is negative, then it's a death-like term. But based on the intuition from what I showed you about the birth-death process, we're essentially assuming that the strength of the noise is overpowering the deterministic death rate here. To the other part of your question where you said, well, why don't you just have additive noise with a long tail? I think I don't understand. So we can talk later, maybe.
Okay.
Yeah.
Yeah, so I'm curious. So you had the situation where you went down to like 100 species, right? When it was in the tail.
Yes.
And so obviously like comparing like the ZIF distributions that you get there to the like, I don't know, healthier cancer situation, is a bit weird. I wondered if you thought about, there's a way where you can collapse the x-axis down onto like, so you have like relative rank and you scale up the kind of frequencies so that it's still normalized. So kind of like a relative rank frequency distribution, whether you think that comparison might yield anything.
That would be weird. That might be illegal. Like it's different than rescaling a distribution because of the transformations that I've done here. So yeah.
Well, you could still do it with the histogram, right? You could
do it with the histogram,
yeah.
For sure. Well, you could do it with the histogram. Yes, exactly. And I think it's precisely because the axes haven't been reflected yet.
Mm-hmm.
Yeah.
Mm-hmm.
Yeah.
Okay. Let's thank again.